So my discussion for today would be about Gramstaining and antimicrobial agents. Under Gramstaining, I will be talking about what is it, its purpose, what type of stain is Gramstain, its procedures and how to perform a Gramstain, and its purpose. And under for antimicrobial agents, I'll be discussing what microbial agent is, how do they work, and its five major targets, and purpose, its types, and its summary. So first we start out with gram staining. So what is gram staining? Gram staining is a common technique used to differentiate two large groups of bacteria based on their different cell wall constituents. The gram stain procedure distinguishes between gram-positive and gram-negative groups by coloring these cells red or violet. Basically, gram staining differentiates what the cell constituents by either gram-positive or gram-negative by knowing its color, whether it be stained red or violet. What does gram-positive and gram-negative mean? So as you can see, the difference is between their color, but there's more to it. Gram-positive bacteria are bacteria with thick cell walls in a gram stain test. These organisms yield a positive result. The test, which involves a chemical dye, stains the bacterium cell wall purple. As for gram-negative bacteria, on the other hand, it does not hold dye, thus they stain pink instead. So you can see between this gram-positive bacteria, it has a cell wall purple color, a stain of purple color because of their cell wall is so thick that they absorb the chemical dye, and thus it's a positive result. And as for gram-negative bacteria, they don't hold dye and they stain pink instead. Again, what type of stain is a gram stain? So gram staining is a differential staining technique that uses a primary stain and a secondary counterstain to distinguish between gram-positive and gram-negative bacteria. Stain cells purple or blue. Mordant makes the dye less soluble, so it adheres to the cell wall, and cells remain purple or blue. So as you can see, I'm just explaining again how gram staining just differentiates between cells, whether it's a cell positive bacteria or a cell negative bacteria, and how the colors would indicate if it's a positive or a negative, and how the dyes work by adhering to the cell walls. So now I will be discussing on how to perform the gram staining, which would be discussed in four different steps. As first would be the prepare a slide smear, then we'll move on to gram staining, which will have more steps, but we'll discuss it later on. Next is we have quality control, and last but not the least is you examine the finished slide under a microscope. So as for gram staining, there are more in-depth process on how to do it. So first is the application of crystal violet or the Hooker's crystal violet to the culture. So you drop about 5 drops of Husker crystal violet to the culture and let it stand for 1 minute. The bacteria here will stain purple. So after that is you wash briefly with water and shake off the excess. Next, you would focus on the application of iodine, which you would also put 5 drops and you let it stand for 30 seconds only, and you wash briefly with water and shake off the excess. Another one is the alcohol wash or the decolorization. In here, you tilt size and decolorize with solvent or the acetone alcohol solution until the purple color stops running. So be careful not to over decolorize when it comes to this step, and you wash immediately within 5 seconds with water and you eventually shake off the excess. Lastly is the application of the safranin, which is the counterstain. So again, you do about 5 drops of safranin O and you let it stand here for 1 minute and you wash it briefly with water and shake off excess. Finally, you examine under microscope at both 400 times power and 1000 times power on oil immersion. That's it. Now we focus on antimicrobial agents. So antimicrobial agents are drugs, chemicals, or other substances that are capable of acting by two modes, either kill microbial or slow the growth of microbes, which is called microbiostatic. So antimicrobial medicines can be classified according to the microorganisms they act primarily against. So antimicrobial agents, we mostly know this for like as medicines where they either kill the bacteria, what is known as microbiocidal, or either slow the growth of microbes, which is known as microbiostatic. So, how do microbioles work? So, antimicrobials work at the cellular level to continually disrupt and prevent the growth of microorganisms by creating inhospitable environment for microorganisms like bacteria, mold, and mildew. Antimicrobials protect everyday products like countertops, toys, surface coatings, textiles, and hospitable equipment. So they have 5 most common mechanisms of action. First would be inhibition of cell wall synthesis, damage to cell membranes, inhibition of nucleic acid synthesis either in the DNA or RNA synthesis, inhibition of protein synthesis, and lastly is the inhibition of enzyme activity. And here, the antimicrobial agents work by having 5 major target processes that differ between bacteria and human cells. Again, bactericidal drugs either kill bacteria or bacteriostatic drugs could either stop bacterial growth. So, what are antimicrobial agents used for or its purpose? Again, antimicrobial agents, it destroys or prevents the growth of microorganisms, which means either biostatic or biosuicidal. It is possible to organize antimicrobial drugs according to the microorganisms in which they function primarily, which we'll be discussing about the different types of microbial agents later on. And antibiotic drugs are used against bacteria, for instance, and antifungals against fungi are used. Again, we will be discussing it on the later on. So, there are actually various types of antimicrobial agents. This can either be a physical agents or a chemical agents. But for now, we will be focusing on the different types of microbial drugs that are commonly available. First off, we have the antibacterial drug. This drug is used to inhibit the pathogenic activity of bacteria, such as an example would be Zithromax. Next, we have a drug that is used to prevent the fungal activity in the host. These are the antifungal drug. An example is miconazole. Another drug which is used to stop pathogenic action of a virus is called antiviral agents. An example would be Tamiflu. And lastly is the antiparasitic drug. This is a drug that is used to prevent the growth of pathogenic parasites. An example is the anthelmintics. So here we classify the drugs by their actions on how they inhibit the activity or prevent any activity of the drug, and you can either get its name with its suffixes such as bacterial drugs for bacteria, fungal for fungi, viral for viruses, and parasitic drug for pathogenic parasites. So, in summary, antimicrobial agents act against bacterial infection either by killing the microbes or by arresting its growth, killing the microbes, meaning microbiocidal or microbiostatic. Then they do this by targeting DNA and its associated processes, attacking metabolic processes including protein synthesis, or interfering with cell wall synthesis, and etc., which are the 5 major target processes on antimicrobial agents. So that's all for today. I hope you enjoyed and learned something.